Welcome to this video about Hot Start PCR. In this video, we'll explore what Hot Start PCR is, how it works, and how it can be used to detect disease. We'll also discuss some of the advantages of Hot Start PCR and the current state of the field. Hot Start PCR is a kind of PCR method, so before you going through this video, you'd better understand the meaning of PCR. Luckily, we've made videos related to these topics before, so you can watch these videos in our channel first. If you've already watched these videos, we can start right now. As we have talked in the previous video, we already know that PCR is polymerase chain reaction. It's a laboratory technique that's used to amplify and detect a specific segment of DNA or RNA. This technique is highly sensitive and specific, which means it can identify even small amounts of genetic material, like viral or bacterial DNA, in clinical samples. So what is Hot Start PCR? Hot Start PCR means that PCR could only start working when there is a proper temperature rising to trigger the PCR. But why do we need a proper temperature? Because through these additional methods, Hot Start PCR is able to decrease the amount of non-specific amplifications which naturally occur during lower temperatures. Non-specific amplifications is a currently occurring situation in PCR. It has the potential to be triggered by forward and reverse internal primers, and the relatively low reaction temperature induces the secondary structure via primer-primer interactions. Such non-specific amplification often complicates data analysis and can even lead to a failed experiment or test. To address the problem of non-specific amplification, Hot Start PCR methods have evolved which inhibit the activity of Taq DNA polymerase at room temperature. So how does it work? A common problem that results in spurious product formation is associated with the assembly reactions at room temperature or on ice in a non-hot start format. During the initial ramping, raising the reaction temperature to 94 degrees Celsius needed to achieve initial melting and then to initiate the cycling. The temperature of the sample obviously must pass 72 degrees Celsius on the way up to 94 degrees Celsius. At 72 degrees Celsius is the optimal temperature for primer elongation for Taq polymerase. Non-specific PCR product can be generated even if the primers are imperfectly base paired to sites on the template where they would not have base paired under more stringent conditions. This is especially true of primer-dimer formation. Any non-specific product made at this stage is more than likely to be co-amplified with the intended template throughout the reaction. To preclude this difficulty, Hot Start PCR is specifically designed to keep the enzyme inactive until the temperature of the reaction is at least higher than the melting point of both primers. In so doing, the incidence of mispriming is drastically reduced, if not eliminated altogether, which would otherwise lead to spurious PCR product formation. This is so important that most investigators routinely use a hot start technique with all PCR reactions. The most prevalent hot start method involves the use of an anti-Taq antibody which inhibits the polymerase activity of the enzyme until the antibody is heat denatured. The development of anti-Taq antibodies was a significant milestone, improving the quality of PCR data significantly. Commercial preparations are widely available under various trade names. Alternatively, polymerase activity can be transiently suppressed by associating it with an aptamer, i.e., a short nucleic acid molecule with a protein binding ability. The aptamer mediates Taq polymerase activity by transiently binding to it in a temperature-dependent manner and preventing it from synthesizing non-specific product at lower temperatures. Other hot start approaches that were once fashionable including manual hot start, ampliwax, magnesium plus beads, and other methods have fallen out of favor due to the anti-Taq antibody innovation. The application of hot start technology is an important facet of any RT-PCR based assay and can be the deciding success factor in assays featuring difficult templates. However, there are still some methods being used. Aptamer mediated, where Taq polymerase function due to aptamer dissociating from the enzyme at a lower temperature and shorten protocols by eliminating the need for a specific heat activation step. Or the incorporation of specialized hot start DNTPs into protocols. Modified DNTPs with thermolabile protecting groups dictate when the reaction will begin. Hot start PCR offers many advantages. First is minimizing nonspecific amplification. Second, convenient room temperature reaction setup. Third, prevents mispriming and primer-dimer formation. Fourth, appropriate for specific annealing of the primers and their extension. Fifth, high yield and specificity of target amplicons. Sixth, wide range of amplicon length up to 10 kilobases. Seventh, suitable for high throughput applications, including automated liquid handling platforms. And 8, enhanced data reproducibility. As it has so many advantages, Hot Start PCR can be used in any PCR. Hot Start PCR can be used in any scenario where PCR is applicable. When researchers or doctors require accuracy, add anti-TAC antibody into the test is always a choice. Anti-TAC antibody can be brought from companies like Thermo Fisher or Vitria, or you can buy polymerase product with antibodies. Hope you enjoyed the video, and see you next time.